Did you know that in some species, the temperature at which an egg develops determine whether it hatches as male or female? Sex determination is the foundation of life's diversity. But how does biology decide whether an organism becomes male, female, or even something in between? Sex determination refers to the biological mechanism that defines the sexual development of an organism. Simply put, it decides whether an individual develops as male or female, or in rare cases, a mix of both. The term sex here refers to the biological and physical characteristic related to reproduction. But what drives this determination? The answer lies in the complex interaction between genetics and environmental factors. In this video, we'll explore the genetic basis of sex determination, including systems like XXXY, XYXO, ZWZZ, and Haplodiploidy, as well as environmental sex determination, where factors like temperature, social structure, and even pollution play a role. Sex determination can be broadly classified into two mechanisms: genetic sex determination and environmental sex determination. The genetic sex determination is the process where an organism's chromosome decides its sex, and environmental sex determination is the process where external environmental factors influence the development of the sex. Let's get into genetic sex determination. It is controlled by specific arrangement of sex chromosomes in an organism. The sex of the individual is determined by specific genes or chromosomal combinations. It involves the inheritance of sex chromosome that controls the development of sexual characteristics. Different species have evolved unique systems to regulate this process. Let's see XX and XY system. In this system, female have two identical X chromosomes, that is XX, while males have one X and one Y chromosome, that is XY. This is called male heterogamety. Here, male produce two types of gamete, one with an X chromosome and one with the Y chromosome. The key player here is the SRY gene on the Y chromosome, that is, sex determining region on Y, which initiates the development of male characteristics. In the absence of Y chromosome, the default development pathway is female, with the formation of an ovarian and the development of female characteristics. This system is found in humans and many insects. Next is XX and XO system. In this system, the female are XX but males have only one X chromosome and no Y chromosome. There is no Y chromosome in this system. The absence of the second sex chromosome determines maleness, and the presence of second X chromosome determines the female. There is no Y chromosome, and instead of diploid males, they are haploid. That means they have only one set of chromosome. And this type of system is observed in grasshoppers and some moths. Next is ZW and ZZ system. This system is reverse of the XX and XY system. Female are heterogametic. That means they produce the two types of gamete, while males are homogametic. The W chromosome contains the gene responsible for female development, while males develop from two Z chromosomes. The presence of W chromosome triggers the development of females. This system is found in birds like chickens, some reptiles, and fishes. Next is Haplodiploidy. In this system, males are haploid. That means they have only one set of chromosome, developing from an unfertilized eggs. Fertilized eggs are diploid. They develop into females, while unfertilized egg, that means haploid, they develop into males. Sex determination is influenced by whether an egg is fertilized or not. In species like honeybees, ants, and wasps, sex is determined by the number of chromosomes. Next we have environmental sex determination. In this mechanism, an organism's environment determines its sex. Unlike genetic sex determination, there is no fixed genetic instructions for the sex development. Temperature dependent sex determination. Temperature at which the egg incubates determines the sex of the offspring. The specific temperature range that produces males and females varies by species. Type 1. Higher incubation temperature result in females, while temperature which are higher result in males. Simply put, cooler temperature produces males and warmer temperature produces females. This is seen in turtles. Type 2. Lower temperature results in females, while higher temperature result in males. It is just the opposite of type 1, and these are found in crocodiles and alligators. Type 3. Some species show intermediate temperature that leads to female with extreme resulting in males, and this is observed in some lizards. This adaptation often ensures population stability in changing environment. Next one is social environment and population density. In some species, social structure influences sex determination. For example, in clownfish, the largest individual in a group become the female, while other remains male. If the female dies, the next largest male changes sex to replace her. That means if the dominant female dies, the dominant male will change the sex and become the new female. This ensures breeding pair mismatch. This phenomena is known as sequential hermaphroditism and is an adaptation to maximize reproductive success. Next is pollution and contamination. Exposure to environmental pollutants like endocrine disrupting chemicals, which are DET, PCBs, they can interfere with normal sex determination process, causing squid sex ratio or even the production of individual with both male and female characteristic, that is hermaphrodites. These pollutants can interfere with normal system during critical developmental period, leading to changes in the sexual differentiation, and these are seen in some fishes, amphibians, and reptiles. Sex determination showcases the incredible diversity and adaptability of life. Whether driven by genetic or environmental factors, it is a reminder of how finely tuned and complex biological system can be. That's all for this video. Thank you.